Today we've got Elena from Harry Perkins talking with me, and we're going to be discussing something which I've been really fascinated with for the last few years, which is how data science is changing high-performance computing. I myself am a physicist, so I'll go into that. So I'm Stuart, I'm the CIO here at Doug. I've got a PhD in quantum physics. I spent many years writing a lot of computational code and running it on the biggest systems in Australia. A very long time ago though, some 20 plus years ago. I've actually surprisingly hidden some of my results in this talk. I'm from this company called Doug. So who are Doug? We're a service company at heart. Right? So we've emerged from the oil and gas industry. We've been building some of the largest commercial supercomputers in the world now for some time, and we've been writing this very large, sophisticated software package. Underneath all of that, we have this large R&D organisation. So we've got some 40 plus people in this building who write our software and develop our tools and help us run the computers. Everything sort of below here, I think, is pretty safe to say it's nerd territory. Right? We're all mathematicians and physicists and engineers and geophysicists and all of these sorts of things. A large percentage of our company, I think it's something like 30%, have got PhDs in A science. So it's very top heavy. We sell all of this as a cloud. Okay, so people can come onto our cloud and use high performance computing as a service. They can use us for services where they give us the data and we process it for them. To do this, we have some 200 plus data scientists in the company. So it's quite an extensive group. As I said, we write software, we have a very large sophisticated package, and we sell it all as a cloud. And then we have Elena, who's going to be speaking with me today.
Right, hello everyone, my name is Elena. I'm a bioinformatician. I have about 10 years of experience working in industry and in academia and different projects. And I got my PhD from University Auckland in 2018, and it was in genetics, but actually bioinformatics. So at the moment I work at the Harry Perkins Institute of Medical Research in Netherlands here. And institute does all sorts of things related to medical research. We have a dozen or so labs that do different aspects. There are some engineers, there are some people who do molecular sciences, but in general we are trying to conduct innovative research that translates into lasting health benefits. So today I'm going to talk about bioinformatics, and one first question I guess people ask most of the time is, what is bioinformatics? So bioinformatics is a multidisciplinary field, and it's a wild mix of mathematics, statistics, computer science, and also surely biology. What bioinformaticians do on a daily basis, there can be completely different things, but we can classify maybe roughly in these three categories. So we can develop novel algorithms and methods and models, we can develop novel software, or we can do data analysis and interpretation. But I guess typically people end up doing a bit of everything, at least to some extent. Myself, I'm mostly sitting in the last category here, but naturally I have to do some of the other things as well. Bioinformatics is also a very diverse field, and there are plenty of things bioinformaticians can study. This can be, for example, these are just some examples, can be studying just sequences, genomic or proteomic sequences. We can be looking at two-dimensional, three-dimensional structures, or we can be looking and working on the kind of highest level of organization, looking at our cells, why our cells are different, even though they all start from the same genomic material, and how cells end up interacting to each other, how cells form tissue, what's normal functioning of the tissue, and what happens when we get diseases. At the moment, most of the time I work with single-cell RNA sequencing data, and this comes from the very special technologies that was actually developed not so long ago. So with this, what we do from the experimental point of view, we take chunks of tissues. In the lab, we mostly work with cancerous tumors, but not necessarily. We have some other projects as well. So we get these chunks of tissues, we dissociate them to release individual cells, and then all these individual cells get sequenced, and we get this genomic information from them. After that, from the sequencer, we get raw data, and the raw data comes in the form of BCL files that looks just like hundreds of files, of binary files, and it adds up to about 100 gig for 8 samples, but it also varies depending on the experiment you do. There is another type of data we also consider to be raw data. It's called Fastq files. So Fastq files, they are kind of secondary in a way because they're derived from BCL files, but they are a bit more human-friendly and a bit more human-readable. They look like this. There is a string of letters, that's our sequence, and there is also lots of sequence-related information, such as, for example, quality. And we get, there can be different number of these files, but they add up to about 10, 20 gig per sample. But at this stage, we still can't really use them. They're still not particularly meaningful to us. And in order to make a bit more sense from them, we first need to do some data pre-processing. With this type of data, as in single-cell experiments, we do first read mapping and counting. So we are figuring out which genes these reads came from, and we do some quality control and filtering. But in the end, we end up with matrices, which are quite already informative at this stage. So we have, for one sample, we can get around 20,000 genes as rows and 6,000 cells as columns. And one of the ways to store these is this is called MTX format, just 3 different text files per sample. And once we have this, we can already start actually the analysis, and we can start asking and answering questions. There are multiple different types of analysis, like dozens of things that you can do. Here are just some of the examples. So you can do, quite commonly, we do clustering analysis just to identify similar or dissimilar cells. We can do cell annotation, so that's very important for single cell field, because when you sequence stuff, in most cases, you don't really know what you sequence. So already once you got the data, from this data, you need to actually figure out what you just did. The next type of analysis is differential gene expression. That's also quite common, and that's when we, for example, want to compare things. We can compare different cell types, or we can be comparing different conditions and looking at what genes differ between those. So, for example, what genes are responsible for drug response or for making certain patients not responding to a drug. And there are many more types of analysis. I guess one of the things that is common for many of those types is that there is no really, for us, opportunity to just run it quickly and then be happy with the results, because most of the analysis and most of the programs we use, there are plenty of different parameters, there are plenty of different ways you can run the same more or less tool. So what we end up doing is running the same type of analysis in parallel multiple times with different parameters, and then trying to figure out what works best. And so the type of analysis, it surely depends on the biological question that was asked in the experiment. But even for the same biological question, you will typically have dozens of different software packages to choose from, and that adds additional challenge. So, for example, you can see here a couple of plots from this database, which collects tools for single-cell RNA sequencing data analysis, and they're tracking already almost 1,000 tools. So that's just, again, just for single-cell data analysis. That's a really, really narrow field in bioinformatics, and it's also relatively new. You can see it starts at around 2016, and there are already 1,000 tools. So that adds complexity to our life, because we need to keep on track with all of this and try to all the time be up to date with what's happening and try to find the best solutions. And another complexity here is that these packages are often written in different tools. Some of them were not initially designed for creating multiple, like, complicated pipelines and packages. And they're all also quite often written by people coming from completely different backgrounds. So it's not necessarily software engineers, it's not necessarily packages that follow the best practices. So that's also part of the challenge that we have to work with.
Cool. as I discussed, I come from a physics background, and people often ask, well, what is physics? And it's a natural science that studies matter, its motion, its behaviour through space and time, and the related entities of energy and force. I guess to a physicist, everything is a subset of physics. An important point that I try to make to people about physics is that physicists develop nearly every bit of technology we use in our everyday life. Doesn't matter, lasers, microwaves, phones. I heard someone say that there's about 30 Nobel Prizes in physics in a mobile phone. So it's used absolutely everywhere, even though you don't know it. Physicists generally invented the computer. Here's a photo of the first silicon-based transistor. And Intel's founders were physicists. One was a dodgy physicist, he was a chemist physicist, but they both have physicist in their name. And I guess the important part of what I'm trying to point out here is that physicists have been running code on computers since they were invented. Okay, physicists have been there and evolved with computers in an unhealthy possible way. Okay, so just keep that in mind, everything I talk about, physicists were there at the start. What do people use physics for? Look, atmosphere and ocean modelling, earth modelling, fluid flows, quantum systems, molecular systems, optical systems of the universe, and processing data from experiments. And what we have here is a photo of a high Reynolds number fluid flow through a pipe, and you can see the turbulence. And this was a job we actually ran on our system. We ran it simultaneously across about 1,000 nodes. So it was a very large memory, very, very complicated, detailed study of what happens with fluids when they go really quickly through pipes. But physics tends to be driven by a theory, an idea, and a mathematical model. So you basically propagate something through time according to a set of rules which are derived, are physics-based rules. They don't tend to be looking for something we don't know about in some data we don't know much about. Okay, so it's not generally looking through data. It's generally starting off with something and modelling forward, or taking data from an experiment, applying a heap of physics rules to that data, and coming out with an answer. Physics is what I call well-structured. Known mathematical procedure, well-ordered grids. Everything like these photos are on grids. Generally simple grids. Compared to your database, there's generally a small number of codes. This is slightly funny because if you go into physics, there's probably a bazillion codes. Each academic writes their own, but in terms of packages out there on the internet that you can download and run, there's only a small number. They tend to have simple workflows. Put some data in, run a mathematical model for 6 months, come out with an answer, and there's typically only one answer. There's not different ways you can construct. You can have different assumptions in your code, which will give you different results. But at the end of the day, you could run the same input with the same code and get exactly the same answer. Okay? So this leads to well-structured use of high-performance computing. As I said, high-performance computing is everywhere in physics. It's been around for 70 plus years. All physicists learn programming and logic. If you go down to Curtin and UWA, they both, in their physics departments, have high-performance computing courses. So at the undergraduate level, you are learning how to use high-performance computers. And there's a long heritage of users. As you can imagine, physicists have been doing computing, running their models for 70 plus years. Lots of people in the physics community do it. Interestingly, this last point is perhaps a very important one. A lot of physicists end up working in supercomputer facilities. I'm one of them. Right? I did my PhD in quantum physics, and then I moved over to sysadmin and running supercomputers and helping users. So for physicists, there's plenty of support. When you are a physicist on these machines, chances are the person you're asking for help from is also a physicist and understands your mathematics. Right? So there's lots of implicit support in our current system. But times are changing. And this is what I'm finding really interesting. Things like the Large Hadron Collider and the SKA are massive experiments collecting massive amounts of data. And it is starting to look a lot more like data science. It's starting to look a lot more like bioinformatics. They're applying a lot of artificial intelligence, machine learning, deep learning. As I said, it's looking a lot like bioinformatics. Trying to find stars in this starts to become an image processing type problem. So I guess I'm really interested in how high-performance computing systems are adapting. I think we've seen SSDs delivering a lot higher performance, especially for the data-driven sciences. Products that we use like VAST make a huge difference. We're seeing the software environment evolve really rapidly at the moment. And this is, we're seeing containers, they popped up only a couple of years ago and they now seem to be dominating certainly what the bioinformaticians do. They use a lot of containers, they use a lot of software repositories. There's all these workflow management tools appearing. The hardware is evolving as well. Before we used to just have CPUs. Now we have TPUs, GPUs, APUs, NPUs, IPUs, QPUs, DPUs, and VPUs, and no doubt more. All right, so these hardware are all doing different things. They're all optimized for different problems. None of them are generally optimized for physics. Physicists work out how to use them because the machine they're going to use has got them. But generally, the things like the TPU from Google are explicitly targeting AI. So again, the hardware is evolving, new products are appearing, and software is becoming incredibly important. So of course, the physicists are feeling a bit miffed, so they thought, bugger that, we'll go and invent quantum computers and just blow everything out of the water and start from scratch again. So I guess this sort of conversation we'll be having again in 20 years' time when everyone's got a quantum computer on their desk and everyone else is starting to shift over to them. So that's coming, which will be interesting. But I guess in the short term, Elena, I have a few questions for you. So, as I've been in high performance computing for a little over 25 years. When did you start?
Oh, so for bioinformatics, I started about 10 years ago, but HPC, it's been on and off depending on the project. So I can't call myself like consistent user of HPC.
Cool. And I guess which parts of high performance computing and data science do you think are troublesome for bioinformaticians?
Here with bioinformatics is a bit hard, depends really on the users, because depends where people come from to bioinformatics. Because it's relatively new field still, and some people actually come from just biological background. It often happens, for example, at the Perkins, happens a lot where people perform the experiments, they know how to analyze the small amount of data, but then the needs of the lab evolve, and they need to do now high throughput experiment, which yield lots of data, and then they want to start processing this data themselves. But they don't really have necessary background, so they maybe use just Windows their whole life, they never try to program anything, and they never try to use command line, and all these black screens, they actually scare them a bit in the beginning. So these, at least from what I see, that's kind of the largest struggle people have. And another thing here, there is also not that much support for us, I guess, because, for example, take again as an example where I work, so we obviously have IT department, which is actually at UWA, but IT department works with standard users who have Windows laptops and who have problems connecting to the printer, for example. Whereas if we have problems with servers, IT tells that it's not something they are happy to help with. So that's another big struggle, because we are just on our own. No one's there to help us.
Yeah, indeed. And I guess what are the pathways into high performance computing? I mean, bioinformatics is something which is coming up, but how do they get into high performance computing? You've touched on a couple.
Yeah, so that's a few things already mentioned. You can come from biological background, and you can come from also mathematical background. So in this case, people would have maybe some experience with programming, maybe some experience with use of resources, but then they learn the other side of things. They learn our science, they learn the biology. Yeah, and also, as I already mentioned, it often happens when you just need to transition to get more resources. You maybe even work normally on some remote server, but now suddenly you need HPC to do some more data analysis, because also each year technologies develop in these lab sciences. Technologies develop super fast, and every year we get more and more data, and every year we need more and more resources. So soon, I guess, everyone will just have to have access to HPC.
That's quite interesting. I mean, I look at Doug's seismic processing business, and the mathematics has been around for 60 years, 70 years. Would you geophysicists agree? It's been around an awfully long time. It's just we have never had the horsepower to do it. And I guess that's one of the interesting things about physics is that they've sort of grown up programming in their undergraduate courses, and then they get to a research point and the problem's just bigger. You're going to run higher density. You're going to run, instead of a grid that's 1,000 by 1,000, you're going to run 10,000 by 10,000 by 10,000, add another dimension, stop doing two-dimensional stuff. So it's quite a different pathway into high-performance computing. And I guess the other interesting thing, which I alluded to, is the data is different. So how do bioinformaticians use data?
So in our case, data comes from the experiments always, and it can be multidimensional. It can be a lot of information just from one experiment, and then we get maybe one experiment per week. And what we need to do with the data, we need to extract information from it in the long run to inform, for example, the medical decisions or to answer biological questions. But data also needs lots of investigation, so we need to actually understand, based on how the data look, how we should treat it in order to extract the information. That's, I guess, different a lot from the first from what.
Yeah, I mean, as I sort of pointed out in this slide here, I mean, we start off with a mathematical procedure, start off with an initial condition for your grid and you propagate it or something. So again, quite often what we do as a physicist is model driven. It's not necessarily data driven. Again, I find really interesting. The impact on a high performance computing system is fascinating because you know, this was actually my work. When you generate these plots, you literally at the end, you run your program for some length of time, and at the end you dump out this regular rectangular grid onto disk. Whereas when the bioinformaticians have thousands of files, all ones and zeros, or A, C's and G's, and I don't know what else is there, T's, and trying to mix and match, match them and do all sorts of things. It's a very different data access pattern as well. As I sort of alluded to, the SKA and the Large Hadron Collider are generating colossal amounts of data, and just the process of trying to manage that data is becoming a huge data scientist project. And I think it's keen that we talk about that in Data Science Week, because a lot of data science isn't necessarily about extracting value of data. It's also just learning how to manage the sheer volume and extract the metadata and keep the metadata in databases, which I guess bioinformaticians must do a bit of.
Yeah, so in terms of data management, we kind of learn as we go, because most bioinformaticians don't have clear data analysis background. So you kind of learn on the job, and then again, as a result, you don't necessarily follow the best practices. You kind of learn by yourself, okay, what exactly you want to back up, and usually we also want to keep everything. We want to back up all our raw data, hundreds of gigs. We want to keep all our intermediate analysis results and all the intermediate files as well, and obviously the results. So that's also, I guess, quite different from what you would do.
It is. I mean, most physics stuff, the input data is trivially small. And you get lots of data out. And actually, I think in terms of my own PhD from 20 years ago, I ran the code the other day for the first time. Compiled straight away, fed the input in, and I can generate the results. What took me months on a supercomputer 20 years ago, you can now do on a decent sized machine in just mere hours. So it was really, 20 years on, you wouldn't store the data. You would just regenerate it, because you can regenerate it exactly. Whereas that's not the case with your data.
Yeah, quite often, even if you try to use the same software, the same version, you try to install it the same way, but it gives just different results. In some cases, there is some stochasticity involved. In some cases, you just can't manage to reproduce exactly your environment. So yeah, we prefer to keep stuff rather than to try to reproduce it later on and see that you get actually different results and you can't really publish your manuscript.
Yeah, which I guess drives the pickup of containerization and workflow management programs like Jupyter, because you actually have a complicated workflow that you do want to reproduce for a paper or a scientist in the future. So I guess just to sort of slightly wrap it up a little bit, I don't know where we are with time, we're getting close. What would you like to see change in the high-performance computing? If you could tell us, as a high-performance computing vendor, to change something or install something or build something different, what would you want?
Yeah, well, I could tell from the point of view of the users, as probably for people who do more data analysis and interpretation, that we want to spend most of our time analyzing the data, not really installing stuff, not really worrying about how to organize hardware, what to install, when to install, and do all this troubleshooting. So we want to just analyze the data as much as possible. We also want to be able to switch between different versions of the software. Conda actually solves this problem, at least partially, so that's already good. And we always want everything to be as fast as possible, but that's, I guess, not surprisingly, right? Yeah, and one of the things people, I guess, get a bit frustrated that they can't really use easily with HPCs, graphical user interface, because if people come from biological background, that's something they're more used to. And then when they switch completely to the command line, it's a bit hard. So that's, I guess, the wish list.
No, I mean, it's great. I mean, I guess as a facility, the team up the back will attest to this, where the growth of interactive high-performance computing and being able to use desktop-type environments and have the back end run is becoming an ever-increasingly important part. Certainly something we've seen a lot of. For myself, I can't look at it easily, but certainly the Jupyter notebooks are something which we are seeing more and more of. It's interesting. And the package management environments, I mean, you have Conda is one, but there's a plethora of package management systems, and even just knowing which one to turn to for your discipline is really interesting. And physicists like myself, you just have your code that you've built over 20 years and compile it and run. We worked on Igor Bray's code from Curtin University, and you dive into his code, and I was just looking at it, and my head wanted to pop from this very old Fortran 77. And he said, some of this code I wrote in the 60s, and it still compiles and still runs and still generates the right results. The results that he wants. So it's fascinating, just a completely different way of thinking about the problem. So, questions from people in the audience before we...
So my question's for Elena. Most of the software on that website, you said like there's 900 tools or something.
Yeah.
Are these open source or proprietary?
Yeah, mostly open source. Bioinformatics is moving to pretty much everything, I guess, is open source.
Mm. And mostly command line?
I wouldn't say mostly, no. But yeah, a big chunk is command line. I guess for people who want really proper graphical user interface and really proper kind of interactivity, there are some commercial versions. But sometimes what people do with the commercial software, they actually take the open source one, fix it, improve it, make it pretty, and sell it. Mm. So that's actually one of the ways it happens.
With so many bioinformatics tools there, how often do you find, well, amongst your colleagues, don't have to name names or anything, people updating which package they use? Or do they tend to stick with what they know? Or do people go hunting for the latest and greatest and the fastest or new features and things like that?
Yeah, it's a constant struggle, and you always try to reach the balance. You don't want to spend all the time trying to look for better options, because then you don't have time to publish. But you don't want to use something from 3 years ago that's already obsolete and maybe even proven to be wrong. So you always kind of try to balance this. There are a few packages now that are starting to be more and more popular. So once people stick to this, it's really hard for them to switch. But in general, I guess, yeah, from time to time, everyone goes out there, says, okay, I'm going to try these few new packages, tries them, but then in the end might as well go back to the favorite.
Thanks.
I know there's a push in Australia, at least in the academic place, to have this, what's called fair access to data. Is that becoming popular in bioinformatics?
So with the data, now when you publish, there is an incentive for you to upload raw data to the open repository. And there is an incentive to upload scripts as well. with the data, it's kind of a bit more compulsory. So in some cases, if you don't upload the data to some of these specialized databases, you don't get to publish. But with the scripts, it's still not really forced. So quite often you can get the data, you see the methods that you, in theory, can reproduce. But because the authors haven't uploaded the scripts, when you try to reproduce the methods, nothing works.
Interesting. Any other?
Just now you said it is difficult to reproduce the result,
Yeah.
even if you try very hard. What happens if you run the same thing twice and you get two results? Which one do you trust?
Yeah. So, well, so these kind of problems, they start happening when you either update. So for example, if you work with your own analysis and you update software, then you get new results. Then you kind of hope that they updated in a way that they removed bugs and made it better. When you try to reproduce the paper and it doesn't work, then it's just completely different story, because in most cases, it's just the authors don't really provide enough information on how to reproduce it. But in general, you also get this problem when you compare different software packages, because they all give you different results. And because you don't really know what's the right answer, it's really hard to choose. It's close to impossible, because in some cases, you have kind of gold standards, and you can run these tools on your gold standard data set and see what works best. But that's, I guess, just small percentage of problems like this, and in most cases you just try to see what makes most sense biologically. So there are certain things that when you get the results, you understand, okay, that's 100% not true, that doesn't make any sense. But yeah, it's a lot of kind of trying to find the right answer without having really enough information.
That's really interesting. I mean, I know in a lot of what we do here in Doug, and myself personally in the coding I've done, you basically get theoretically known results. In my space, if you don't have a potential in the way and you propagate a particular shape, it stays that shape as it propagates. And so after you propagate a while, you know theoretically where it should be, and you can check your results, at least for a small number of theoretical cases. It sounds like that's not the case in your field.
Mm-mm. Yeah, no, because what we are doing, we are trying to, so for example, in my, in the work that I do, we are trying to find biological answers for biological questions, and we don't really know them, right? So we don't really know when patients have cancer, we don't really know what exactly went wrong, so that's what we're trying to do. And it is hard when you get different results from different tools, and then you just try to, and that's why it's bioinformatics, because you also will use a lot of biological knowledge, biological assumptions in order to figure out if it makes sense or not.
Interesting.
So I think that's very common, in the oil and gas industry. If you're doing any kind of calculation which is impacting your finances, you have to keep that software and that version of the software which is used for doing it for perpetuity, or until that's no longer a financial issue to you. So like we have some companies which I've worked with in the past had stuff which was dating back to the 70s, which they had to keep because somebody called them on like how they actually arrived at those figures. That's what they've got to run. And then you have to find hardware to run it. It's like, it's an interesting problem.
Yeah, again, the language space is really interesting. I mean, this is just a small smattering, but R, Python, C++, MATLAB, and then in this other, there's probably just a bazillion other languages. I know in a lot of physics, it's sort of C, Fortran, and then it stops really quickly after that. Probably Fortran's this one and C's like this. It's a completely different space. And as I said, I find it really fascinating how things like Python on our system, if you install it a particular way, just starting up a Python script can take minutes. You change your file system and starting this up can take fractions of a second. So if your job only lasts a minute, then how you install this software can make a massive difference to how quickly you can run it. And it's just little things like that can make a big difference. And I guess in the high performance computing space, More focus is coming this way. Less focus on the C++, MATLAB, well, don't bother with that at all. But the C++, Fortran are probably not quite at their peak, but the systems are set up for those. The systems aren't set up for these yet. And they're getting a lot better, but they are certainly not quite there yet.
And there's in the US, in the national labs, there's a strong push to rewrite everything into Julia, now add Fortran to Julia.
Right.
Which is interesting.
Well, I wish them well. They won't get there. Fortran made a comeback in the last study. It moved out of the top 20 programming languages, and it's moved back in the last few years. So it's actually gaining in popularity. So
why is it getting back in popularity?
probably because people start to realise it's really good at what it was designed for. If you want to work with large arrays, like mathematical arrays, multiply two arrays together and deal with complex numbers, there's not much else really out there. It is very, very efficient at those types of things. It's a very small, simple language. Very simple data structures. You can have a plethora of really complicated data structures in this space and do really sophisticated data management things. But if all you need to do is invert a matrix, Fortran's going to be there real quick.
Sorry, I missed the beginning. Did you guys mention anything about Curtin's hive?
No.
So I've been to a demonstration. They're doing this. They're freezing the plant cells. They're gonna something to do with study of how they freeze the plant cells, plant materials, using different types of alcohol.
Right.
And so I thought maybe perhaps that's more to do with data visualization as opposed to high performance computing.
Yeah, probably.
But they did crunch the numbers using the supercomputer, and then they
Right.
presented.
Interesting. Because I imagine then they'd have to worry about the cells exploding and things like that and destroying the structure. So,
Yeah, they wanted to keep because apparently the plant cells are able to be deep frozen,
Right.
as opposed to human cells.
That's a molecular dynamics study in at the same time as experimental. That's from my group, my supervisor.
So they run the simulation, I guess.
Correct. So what we do, not me but colleagues of mine in my group, is that we study how the different alcohols interact with the membranes
Yes,
that envelop the cell, and that way they can infer why some alcohols at some concentrations, at some temperatures, are better than the others. And this comes back to what we were saying earlier, that yes, in biology, there's so many unknowns, but when it comes down to the interactions of molecules, they can be attractive or repulsive. But once you put a large number of different molecules, and you want to trace what they're doing on average on time, then it becomes a problem of we need to use, the supercomputer to run the behavior of the molecules, and then we need a lot of this to actually visualize and be able to measure little things, distances, predict bonds and predict attractions and forces and energies from it. So yes, it needs a lot of power for the visualization, but it needs way more than that to actually run the simulations. And then there's always the problem of, your system wasn't large enough. How can you be sure of that? Which is again how verifiable it is, because we know what's a biological relevant free energy, for example. But if you're within 10% of that, then you can probably rank another alcohol worse. But when you run the experiment, it's reversed. So it really matters how accurate you get and how well you can predict things.
It was in 3D as well. Was one of the Curtin Uni Research Week, and managed to. That was really good.
Yeah, some really interesting stuff that happens. All right, any other
I'm not sure how to verbalize it, but I have a whole bunch of comments based on Elena's talk. I thank you for your candor in what you say. I hear what sounds like a lot of bad news to me. I'm a petroleum engineer, and I can recognize you describing a lot of things that happened to us when I started in the oil industry. Everything was analog, and we did everything by hand. We didn't even have typewriters. Then we went into computers. Then we had a golden age of HPC, actually. So in my particular discipline, it was the second half of the 90s, where everybody was getting into it because we could. And there, one thing that happened very clearly is everybody started using tools that were readily available, but that they did not understand. And after using them, they still didn't. And all sorts of bad things came out of that. And possibly as a reaction, I'm not sure, but that was abandoned. So at the end of last millennium, we stopped HPC in my discipline and haven't done it again since. And I suppose the understanding has just gone down and down in the physics of what we do, while we spend all of our time dealing with data and the absence of standards, and the fact that the code that was written by another bloke that doesn't quite work. There was a mention of which software to use and what to do. Every software, of course, has its own idiosyncrasy. So if you learn to work your way around the quirks of a particular software, that's an investment in time which you might as well leverage for all it's worth rather than jump to another one. It's very much personality driven. I'd submit to all the people here that much of what you described here is also the experience of everybody else in their own profession, particularly if you're a knowledge worker. That's some people are gadget people, they're tech happy, they embrace the latest thing, others don't. Sorry, I'm not turning to you, but I am fundamental. I am an IT person by academic training, but yes, that's the old yes. But for some reason, I don't get excited by gadgets. I have other friends who are biologists or even HR people, and they embrace the latest tool, and suddenly they start doing whatever Markov chain Monte Carlo stuff with no idea what it does, but it produces colorful results, and their career goes like this. So to go back to an earlier comment, I'm not sure that's great news, and I feel for you because in my discipline we haven't found a way out of this rabbit hole of dealing with data and the absence of standard or the presence of too many. And things like that. But I was interested to hear that your experience is congruent with mine in a lot of ways, and I suspect with a lot of other people's here. Another thought I had was if the reuse of code is so important as it really should be, I reuse quite a bit of code. The code I wrote 3 months ago, which I haven't used in 3 months, so I don't quite remember what it does. Maybe we should put a lot more emphasis on teaching what in the US they call clean coding and things like that. It could perhaps even be a business to take a code that works and clean it up. Now, what's the point in running something written in Python on your computer? Isn't that completely?
No, not necessarily.
Well,
interestingly,
it's a highly inefficient language.
Yes.
On a super fast machine.
What is really interesting I'll go back to this one. So we're talking, Python looks potentially to be inefficient, except that on the back end of Python, they are exceptionally good at using native libraries on your computer. So that we can hook it up to the Intel Math kernel library. So if you're doing sparse matrix operations, Python can use the fastest for your CPU. Where the inefficiencies come, I find, with Python is when you've actually got to pull data out of those routines and push them through Python, push them into another routine. In a C program or Fortran program, that would probably involve no memory copies and no memory movement, whereas in Python it probably involves about 50. So it's those interactions, I think, where Python falls down. However, when you're in Fortran and you write a matrix and you want to multiply two matrices together, you call it matmul, right? Multiply two matrices together. In Python, you call a library and you hand it to functions. In your Fortran code, the actual arithmetic gets built into the kernel of your assembly code. With this, it goes out to an external function and then passes through a few layers and may end up on a GPU or may end up on a CPU. So I think there are some real efficiency gains to be had here. It depends on how you express your code. If you can get by with standard libraries and things like that, then there's no reason why this should be slow. It doesn't have a very complicated memory model. It's not very sophisticated in the way it represents mathematical structures like arrays. Like, you really can't do it. You can't talk about complex numbers in this. So these all get hacked in, and that's where I find a lot of the inefficiencies come. Whereas they're built into Fortran. They're just part of Fortran. Complex numbers are just there. And for a quantum physicist, that's everything.
One of the biggest efficiencies is also the human time though as well. Like, to get up and running with Python is a lot quicker than, like, for my code, if I was running in C++, I probably wouldn't have had it run yet. But in Python, I can get it up and going fairly quick.
That's the point about you writing something in Python and then somebody else maybe writing it in Julia or whatever's faster.
Yeah.
In the early days of physics computing, we wrote things in Fortran, and then we put them in machine language. And if it was really good, then we burnt it in silicon. Now anybody can write anything in Python.
I mean, inside Doug, I mean, we take C code, we have a lot of C Java code, and actually the absolute critical components do effectively end up in assembly.
All right.
An interesting thing about bioinformatics is that the actual data collection, the sequencing and stuff like that, that technology is still evolving. And so new machines are coming out that actually change the fundamentals of the data. It comes in, traditionally there was a high error rate in every single value that came in. You didn't know what the fundamental truth was. So initially there was a lot of work trying to do basic statistics, trying to solve what is real, and a lot of this oversampling kind of effect to try and solve that. And in the early days, there wasn't a lot of mix-in from computer science theory into what the biologists were trying to do. The biologists knew we've got all these things here, but we know we're going to try and match these up, and they would do these sort of rudimentary matching and where you can stack these things out and say, well, I've got 5 the same and 1 different throughout 1 different. Simple algorithms like that. And then they brought in some serious computer science, and that's where bioinformatics really got its launch, to actually do statistical analysis on these stuff and different methods of sort of approximate hashing and things like that to try and match and correlate these things in a much more robust way. And so those tools have had to evolve with every change in the technology to acquire the data. And on top of that, then we have, I think, people like Elena and Harry Perkins' group who are coming up with new ways to use the sequences by tagging and introducing different, effectively, ID numbers throughout the process to label different pieces of the results and then be able to distinguish them later. So all these tools don't exist yet. And so something that's rapid development, like Python, as was pointed out, is essential to actually get functional results. And so that's why you see so much development in that space as well. But it's a different kind of problem. In physics, if you've got some raw input values, maybe you have sort of a stochastic function, so there's some error bar on this. But with every single value, there's effectively an error bar on it. And then because the type of data you're collecting is different, then you're going to need a different tool because you've made all these assumptions and these algorithms just no longer apply. And that's why you see this massive evolution of the software and such a growth in different packages. So Elena touched on that, but she didn't do justice to the complexity of bioinformatics and the things she has to do.
Yeah,
it really
it's
is
just small kind of.
Yeah, it's just tip of the iceberg.
And it's kind of just really small part of what we do in the lab because we do all this Oxford nanopore stuff, which has much higher error rate, and then you have to put this into account. Yeah.
So
we might have one more question, and then we might have a beer.
So if I understand correctly, like the problem with reproducibility, like it's not just your Python is installed incorrectly or you got the wrong packages, right? It's not actually to do with that. It's actually inherent in the problem. Right?
Yeah, there's a good chunk of
Bit
it.
of both.
Or the
Yeah, it is.
Bit of both too.
Okay.
Sometimes you change the version of some Python and things like this, and the results change. I mean, fundamentally, this is software, so it has bugs.
I think, as Elena said, you hope it doesn't change much. Within a couple percent. But again, there's such huge volumes of data. You can't track back to where it went different. Like, why is this one different? I don't know. It could be anywhere in this chain of software.
As a company with our software development team, we would find 3 or 4 bugs in Intel's compilers every year. And that is literally the thing you use to generate the assembler to run on the computer. And if there are bugs in that, everything above it's buggy because everything uses it. We find bugs in GCC quite often. Like literally everything that is software is buggy. Everything's buggy. And then things like compilers are buggy. So from there on up, there are bugs. And you change those, you change results.
And what's the bug in that Intel processor 20 years ago?
And it's not just Intel. All the CPUs have had bugs. All of them now have all got bugs. And they code around them. So, anyway, we might leave it there. Anyone who wants to do a tour, we've got a few people who do. I might commandeer Ross or Mark, if they can, take you through the data center. The rest, everyone else, you're welcome to join us out front. I think there'll be some nibbles and some drinks and stuff. And thank you very much, Elena, for coming along and helping us.